Today, we are diving into one of the most elegant and honestly most important processes in all of biology, meiosis. It's this tiny, microscopic dance that makes sure life is incredibly diverse. And, it's the reason you are genetically, completely, and wonderfully unique. So, let's get into it.
Have you ever really thought about that? Sure, you've got your mom's eyes or your dad's laugh, but you are not a carbon copy of either one of them. So why is that? Well, the answer is in this special kind of cell division that basically shuffles the genetic deck long before you even exist.
And that process, it's called meiosis. It's the biological engine designed to create sex cells, we're talking sperm and eggs, that aren't just genetically different from the parent cell they came from, but also totally different from each other. This right here is the absolute foundation of sexual reproduction.
Okay, so to really get how meiosis pulls this off, we first have to tackle a numbers problem. It really all comes down to counting chromosomes.
Think of your DNA like a set of instruction manuals, okay? Now, most of the cells in your body, your skin cells, your liver cells, all that stuff, they're what we call diploid. That just means they have two full sets of those instruction manuals. You got one set from your mom and one set from your dad. But sex cells, or gametes, they're different. They're haploid. They only carry one single set.
Now, scientists have a really simple shorthand for this diploid state, 2N. You can think of N as just one complete set of chromosomes. So 2N literally just means you have two sets, those matching or homologous pairs from each of your parents.
So the entire mission, the whole point of meiosis, is to get from that 2N state all the way down to just N, a single set. And you can see why, right? Because if it didn't, the number of chromosomes would double with every single generation, and that would be a complete biological disaster. So the question is, how does the cell actually do it? Well, it does it in two major stages. Let's start with the first and, honestly, the most complicated one, Meiosis I. This isn't just a simple split. Oh no, this is where the real magic of genetic variety happens.
Meiosis I plays out over four main phases. During this first big division, the cell's main job is to separate those homologous pairs, sending one full chromosome from each parent's pair into two new cells. But the most incredible thing happens right at the very beginning, in prophase I.
And this brings us to the absolute key for creating diversity. It's called crossing over. This is where the homologous chromosomes, the one from your mom and the one from your dad, they literally get super close and swap little pieces of their DNA. Go back to that instruction manual idea. Imagine taking two different versions and just swapping a few pages between them. What do you get? Two completely new, totally unique versions. This is how brand new combinations of genes are made on the fly.
Okay, so after meiosis I, we've got two cells. Each one now has half the number of chromosomes. But, and this is important, each of those chromosomes is still made of two identical sister chromatids. So the next stage, meiosis II, is a much more straightforward process that's just there to finish the job. Meiosis II actually looks a lot like a normal cell division, something we call mitosis. In this second round, those two cells we made in meiosis I, they divide again. But this time, the sister chromatids that make up each chromosome get pulled apart. And the final result? 4 haploid cells, and every single one of them is genetically unique. So let's just put it all together side by side, because this is the key takeaway. These two divisions have totally different jobs. Meiosis I is the great shuffle. It separates the parental pairs, it cuts the chromosome number in half, and it creates brand new gene combos with crossing over. Meiosis II is way simpler. It's just the final split, pulling the chromatids apart to give us those 4 unique cells. So we've seen the how, but let's just take a step back and really appreciate the why. Why does nature go through this incredibly complex two-part dance?
It all boils down to two huge, profound outcomes. First, it cuts the chromosome number in half, from diploid down to haploid. This makes sure that when fertilization happens, the new baby has the right, stable number of chromosomes. Second, and just as important, it creates genetic variation through that crossing over step. That variation is literally the raw material for evolution. It's what allows a species to adapt and survive over time.
But this beautiful, intricate dance isn't always perfect. Sometimes a mistake can happen, and when it does, the consequences can be pretty significant.
The technical term for this kind of error is non-disjunction, and it's exactly what it sounds like. It's when the chromosomes fail to separate or disjoin correctly during anaphase. This can happen to the homologous pairs in Meiosis I or to the sister chromatids back in Meiosis II.
So what's the result of non-disjunction? You end up with gametes, sperm or egg cells, that have the wrong number of chromosomes. Some might get an extra chromosome they shouldn't have, while others might be missing one completely.
And to make this really concrete, let's look at a well-known example. If this non-disjunction error happens with chromosome 21, it can create a sperm or egg cell that has an extra copy of it. Now, if that specific gamete is the one involved in fertilization, the resulting person will have 3 copies of chromosome 21, which is the genetic basis for Down syndrome. So, from the chromosome countdown to the great genetic shuffle, meiosis is this absolutely remarkable process that makes every single one of us a one-of-a-kind biological event. It's just an amazing reminder that the very essence of who you are was written in this microscopic lottery of chance and combination, which leaves just one question, really. What are you going to do with that incredible, unrepeatable uniqueness?